Zoe Kourtzi, she's professor of experimental psychology, where she uses state-of-the-art computational methods and non-invasive imaging. So here the link with imaging is already there. To better understand complex decision-making and adaptive behavior, learning, plasticity across ages and also diseases in diseases. And today you'll be talking to us about dementia. I'm really, really excited for this talk. Thank you for joining. Thank you, Zoe. The floor is yours. Zoe, I believe you're still muted.
Okay, yeah.
Sorry. Thank you. Yes, we can hear you now. Your screen is up and we can hear you. Thank you.
Wonderful. Thank you. Thank you very much, Marius, for the kind introduction. Yeah, so today I want to talk to you about the need of AI and machine learning approaches to understand better neurodegenerative disease, but also to be able to predict as early as possible trajectories that individuals might take moving away from healthy aging to neurodegenerative disease. And I will be focusing primarily on dementia and Alzheimer's disease. So I'm sure you all appreciate this is a big challenge, a big societal challenge. There are globally 50 million people suffering with dementia, and only in the UK, the cost in terms of healthcare and also social care is already accumulating to more than £25 billion. So a really big problem that impacts our society and also our healthcare systems. So in this space, and given what we know from current models of diagnosis and prognosis, there's a huge clinical need for two main reasons. One, as you can see here, is that there are many different subtypes of dementia, and being diagnosed precisely in terms of the dementia subtype is really critical in terms of survival. Although we don't have disease-modifying treatments in this space, early diagnosis and precise diagnosis in terms of the subtype of dementia that a patient may be suffering from is really critical in terms of planning for the patient, but also for their carers and their families. Another big challenge is with the current methods that we approach the diagnosis of dementia, we have a large proportion of misdiagnosis. With the current studies, we are looking for about 20% misdiagnosis. And dementia is very much impacted by comorbidities. So we might have a patient that goes to the GP because they realize that they have memory problems, one of the main symptoms. And the patient will be given a scale to complete. Their scores might be looking rather low on that scale, and the GP might then recommend further testing for a type of dementia. However, it's quite likely that this patient does not have dementia pathology, but they have cardiovascular disease, or they have even more complex cases of mood-related disorders. They might be suffering from age-related anxiety or depression. And we do have disease-modifying treatments for mood-related disorders and a very different diagnostic pathway. Now, if they are sent home with a potential clinical label of dementia, knowing that there are no disease-modifying treatments increases the patient anxiety. Therefore, they'll come back for another follow-up in 6 months' time. Potentially, their symptoms, their cognitive symptoms, their memory loss might become worse, not because they have memory pathology, they have a dementia pathology, but actually because they have simply grown to be more anxious, more worried, more depressed about a condition that might not have currently drug treatments available. So it's really a very complex space, and a space where we absolutely need all the help we can get from machine learning. And so what we need in this space is transparent and interoperable algorithms that can synthesize different data types for early and precise diagnosis, for precise patient stratification, and based on data that is as least invasive as possible, as early and as low cost in the diagnostic pathway. So patients do not have to go through long pathways, diagnostic pathways, that will only increase their anxiety and potentially their cognitive decline. These solutions based on AI can have a great impact on determining clinical pathways for patients, but also in making clinical trials cost effective by ensuring more precise patient stratification. Now, what are the challenges when we look at AI-guided solutions in neurodegenerative disorders? One of the main challenges is that the data we have from clinical populations or even from research cohort are heterogeneous, and they're compounded by different types of comorbidities. Typically, the data we have in our hands are sparse, they're incomplete, they might be unlabeled in terms of we don't have clinical labels for these patients. And the scales that are typically used for assessing cognitive decline are non-sensitive, while measures that we have, tools that we have in our hands for measuring pathology and neurocognitive health can be highly invasive and costly. For example, a lumbar puncture that measures CSF tau, or a PET scan, a radioactive PET scan, are quite invasive and costly. So these are all challenges that we have to overcome in our strive to make to improve prediction and diagnosis in dementia. And focusing a little bit more on Alzheimer's disease, I'd like to show you, based on what we know in this space, how dementia potentially can be targeted with AI-guided solutions. So what you see here is the well-known Jack curves that actually show that cognitive decline in aging is a process, is a continuum that starts from normal cognitive health all the way to dementia. We have some early signs known as mild cognitive impairment, where it's very unclear whether a patient will be stable and will not progress to any pathology, or whether the patient will fast start progressing to a dementia subtype. And as you can see, if we want to move early in this space and make diagnosis early, we currently could be more successful if we use invasive methods. Less invasive methods, so for example, moving away from PET scans to cognitive testing, where patients simply take some tests or scales, can show evidence of decline, but that's at later stages of the disease. And of course, we want to be able to intervene early, either through lifestyle changes or through currently available treatments that delay the disease progression and improve quality of life for a longer period for patients and their families. So the approach we have taken to try and understand how we can better predict early dementia using machine learning approaches is summarized in this slide. And what you see here is we start from feature extraction. We use the least invasive data that we have in our hands from research cohorts. In this case, we use data from ADNI, a large research cohort of about 1,000 individuals based in the US. And we train a model with many different data types. And primarily we focus on MRI scans, structural scans, and cognitive data. And we model the scans and the cognitive data to extract features that are reliable and are predictive. And then we take a classification, a metric learning approach as part of our machine learning method to classify patients. And from this modeling, we then move into a trajectory modeling approach so that we can identify individualized trajectory and make predictions about prognosis. So as part of this talk, I'll run you through in more detail this framework as an example of what we can do with using machine learning approaches in dementia. So as I said, one of the first things we do is we extract features from MRI scans that are highly predictive of cognitive decline, and we use these based on a partially square regression. Our focus here is to extract biologically and clinically relevant features. We want to make sure that what our algorithms are learning is not kind of some associations between, for example, imaging data and clinical scales that might be impacted, for example, by the fact that data has been collected with different scanner manufacturers or of different field strength. So we go through first this feature extraction approach, and what we have seen is that with this approach, we extract clusters of voxels that show a high atrophy, and they relate significantly to changes in cognitive decline, in particular scores in what's called ADNI-MEM, which is a composite cognitive score that indicates cognitive decline. And what you see here is something that is not surprising. You see that these voxels cluster around hippocampal and entorhinal cortex regions, and these regions are known to be impacted highly and early by dementia. So this is a clinically relevant feature. You can see here we go through a process of auto-sample validation for this feature. So you can see that when we look at the ADNI data that we use to extract this feature is highly correlated with performance in memory tasks. And then you can see we cross-validate these from a different study, the Nimrod study, which is a UK-based study using a different actual scale for measuring cognitive decline. So this is a robust feature that then we can give our learning metric algorithm. And the framework that we use for the machine learning framework that we use is a metric learning, generalized metric learning algorithm. Effectively, the algorithm learns 2 prototypes. Every time that there is new data, the prototypes move further away, and we train the model with ADNI data. And we built a model that contains different types of biological data. So the model is trained with the gray matter feature that I showed you how we extracted from structural MRI scans. Also with beta amyloid, which is a marker of dementia pathology, and with APOE4, which is a genetic marker of dementia. And the classification task that we're given to the model is to look at all the individuals in this cohort study and separate at the stage of mild cognitive impairment, so quite early, before we have evidence of Alzheimer's disease, whether the patients will remain stable or will progress in terms of their trajectory. And we do this within a window of 3 years. So if patients have the same clinical label over 3 years, they're considered as stable, while if during these 3 years the clinical label changes and moves from mild cognitive impairment to Alzheimer's disease, these patients are categorized as progressive. And you can see that the model does actually quite well in terms of its sensitivity and specificity above 80%. This is not, as Mihaela was also saying, at this range of 98%, but here what we strive is not to get the best possible prediction with these models, but ensure that the models are interpretable and ensure that they are based on features that are clinically relevant. And you can see that you can interrogate the tensors of this model. That's what the matrix shows here. And you can see which are actually the features that are more highly relevant for this classification. And you can also see at the diagonals interactive factors So clearly, beta amyloid and the gray matter score are very important for making this prediction. Now, in answering the question of how we could actually make predictions from less invasive data. So could we make predictions if we have patients that cannot even undergo an MRI scan? We looked at this question and we looked at making the same prediction by giving the model only cognitive data. So what you see here is we look at data that have to do with memory, cognitive tests that relate to memory, cognitive tests that relate to executive functions, the ability to attend or the ability to multitask and make decisions, and also skills that relate to depression. And we built a model that is trained only on this data, no biological data, no pathology data. And we asked the model to, again, make the same classification between stable and progressive patients. Now, this is a quite fine classification to make without any pathology data. It's not whether we have healthy controls versus patients, demented patients, which is kind of the standard in the field. It's actually making a very precise classification at early stages of the disease that relates to progression. And what you see here is that the model has very similar, actually, accuracy and prediction when it learns based on biological and pathology data versus on cognitive data. So for this kind of, if you want, initial classification of patients into will patients remain stable versus progress, cognitive data and biological data can be both useful. But moving on from here, what we are primarily interested in looking at individual patients and be able to provide clinicians with a score that will help them identify the right follow-up pathway, the diagnostic or treatment pathway for individuals. So what we do is we interrogate our metric learning models, and we built a scalar projection. We use a scalar projection approach here to actually identify how far patients are from the stable prototype and how quickly they move away from the stable prototype. Through this approach, we can determine a score that we call a scalar projection score or a prognostic index. And then we validate this index against other data. So, for example, what you see here is how this index behaves for individuals in relation to cognitive tests that allow us to measure rate of memory change, of memory decline. And what you see is that the higher this prognostic score that it is, that the higher the decline that we see in memory for individuals. The individual points here are patients that are either progressive in red or stable in blue, and you see that at the high end we have the patients that are progressive clustering in terms of this score will be higher in progressive patients. Now this score is really learned based on either cognitive data or biomarker data. But then we can use this approach to validate this score in completely new data. So for example, what we did here is we took a sub-cohort from ADNI that we did not use to train the model with, and we tested these patients. We used only the gray matter from this separate group of patients and their cognitive test. And we put it in the trained model, and what you see is that for these new patients now, the model does quite well in terms of predicting memory decline from this multimodal trajectory score. And it accounts for 46 of the variance in the rate of memory decline. To further validate this prognostic score, we compare it with other biomarkers. So what you see here is that this score actually relates to amyloid signals that are measured with PET, relates to gray matter atrophy in medial temporal lobe that we know is heavily hit in dementia, and also it relates to APO4 in terms of positivity in this score. Finally, we are very much interested in moving away from early stages of disease to pre-symptomatic individuals. So our aim here is to be able to develop the models in such a way that we can use them as prognostic much early, before individuals have any symptoms. And ideally, if we are thinking the future, models like this could be integrated in health checks, the same way where we check for cardiovascular disease, we could be checking for brain health, we could be checking for neurodegenerative disorders early with non-invasive data and with models that allow us to make these predictions before any symptoms appear. And what you see here is, if we look at different samples of patients that either have Alzheimer's disease or at the early stages, or no dementia, they are healthy, they are controls, or pre-symptomatic cohorts, individuals that have actually shown no symptoms, you see that this prognostic score that we extract from the algorithms from our machine learning approach can actually separate the patients in stable, in this space where the scores are low. These patients remain stable and potentially will not show dementia in the future, will not express dementia in the future, or slow progressive or rapidly progressive. So this type of modeling can be really helpful in the clinic, and it can be helpful very early on if we want to make predictions about individuals before they have any symptoms, and we have validated this with pre-symptomatic cohorts. So just to summarize, and we have a program of work now going into the clinic, in memory clinics. This type of approach can have great clinical utility. The models I've talked to you about today are actually quite shallow models. They are interpretable. One can interrogate the tensors and understand what the different features contribute to. We are also working with more complex models in the space of deep learning. And the beauty of this is that really these models can integrate many different data types, and these multimodal indices that we have now can be then used to generate a prognostic score for individual patients that can massively change the care pathway for future diagnosis. So if patients are, for example, classified as stable, but they still have symptoms, maybe they have other comorbidities, maybe they have mood-related disorders, and can be put in a very different pathway than the dementia pathway, and actually be treated for the disorders that they might have nothing to do with dementia. And also patients that are identified as slow progressive versus rapidly progressive can also, this can also make a huge difference in terms of the clinical pathway. Slow progressive patients can be monitored at home, can be monitored at the community, while rapidly progressive patients may need immediate care and a more invasive pathway. And of course, any treatments that might be slowing down the disease would be more appropriate at more kind of slowly progressive states. Through our PPI work, we have had great feedback in terms of how much difference these kind of trajectories, these prognostic trajectories can make to patients and their families in terms of preparing either financially or family-wise for what potentially is to come. Just to move on a little bit in terms of how also we can use these approaches to understand better the pathology and what we can do in this space potentially with impact to clinical trials. We can also use this modeling approach to predict not only changes in cognitive decline, but changes in pathology. And the next kind of couple of slides, I'll show you how we can use the models to predict future changes in tau accumulation, which is a pathological marker, is the best biomarker at this point that we have for Alzheimer's disease. So what you see here is we use the same approach in terms of a metric learning classification and the projection approach. But what we actually look is at regional changes of tau over time. And we did this with, again, looking at the ADNI data, and this cohort has longitudinal tau. So what you see here is we look at patients that have early AD, and we compare them to stable patients. And by looking at changes in their tau over time, we see that our prognostic score predicts that patients for patients of early AD, that they will accumulate almost 3 times faster tau compared to stable individuals. And you can see that we can actually use the same approach for pre-symptomatic individuals, and that's where this makes a huge difference. People in their 30s and their 40s have no good reason to undergo a radioactive PET scan and potentially would not be interested in doing this just so that they can find out whether in the future they might develop dementia. But by using the modeling approaches we have, we can look at pre-symptomatic individuals before they have shown any symptoms and actually predict whether they will be accumulating tau in later life as a pathology marker for dementia. So you can see the models can do this. And this modeling could have also clinical utility for clinical trials where markers like tau are frequently recorded. And what we see here is that with our modeling approach, we can reduce the size or the sample size needed in a clinical trial by 35% if we look at changes in the rate of tau accumulation compared to changes in cognitive decline. Cognitive scales are not as sensitive, while having a marker like tau accumulation can be more sensitive. Therefore, the sample size can be reduced together with better patient stratification for clinical trials and drug discovery. And similarly, what we see is that through this multimodal modeling approach, we can actually predict more reliably than compared to using one biomarker, for example, beta amyloid. And we can again reduce by about 30% the sample size needed for a clinical trial in making predictions based on this multimodal modeling approach. So I'd just like to summarize. So our work so far has looked in a transparent and quite generalizable and machine learning approach that allow us to define individualized trajectories that are prognostic by integrating many different data modalities. It reduces the risk of misclassifying patients and misdiagnosing dementia versus other comorbidities. And this more precise patient stratification has a clinical utility for the clinic and for patient pathways, but also in clinical trials. And we're very excited to see that we can potentially take these tools to pre-symptomatic populations with the potential of having application into clinical trials for drug discovery early in disease stages, but also to potential prevention through health checks. Our hope is that we can take this from the lab to the clinic, to healthcare systems, to clinical trials as a decision support system that will support through this huge knowledge of the algorithms of different data modalities and large population data. We'll be able to support clinicians to make the right decisions in terms of diagnostic and treatment pathways for patients at the right time. And I'd like to close by advertising a large Alzheimer's Research UK initiative that we are involved in. It's known as the EDEN project, the project for early detection of neurodegenerative diseases. And it's a great also call for collaboration. The project is looking into machine learning approaches that allow us actually to integrate cognitive and biomarker data with digital data. The idea here being that potentially through mobile technology, we can collect in a non-invasive way continuous digital data at very early stages, even before disease, even before symptoms. Integrate these potentially into this type of technologies, integrated technologies into health checks that could make a huge difference for prevention in the future. So I'd like to close by thanking you for listening in and joining today, and thank all of our collaborators, both clinical collaborators, but also all our friends and colleagues in maths, computer science and engineering, everybody in the lab who's making this work a lot of fun, and of course the people who give us the money to do all of this work. Thank you.
Thank you, Zoe. That was an excellent and inspiring talk. I'm sure there are a lot of questions, so please, people, put them in the chat or raise your hand. To start off, I was actually thinking along a line similar of what you touched on in the Eden slide that you brought up at the end. You have a pretty good sampling in these large studies, but you can never go back and say, oh, we should have added this. With the knowledge that you have now, what do you think is the domain that we lack most in these large studies, such as UK Biobank or ADNI? Which domain is probably going to have the biggest impact on the accuracy of your prediction models that we don't currently have access to?
Yeah, this this $1 million question or £1 million question, really. And in a way, this is what we need to be doing in a very iterative way. The models can also tell us which are the most predictive factors, and therefore what data we should be collecting moving forward. And we haven't progressed enough, if you want to know. So, for example, on the approach that I showed you, I think I said at some point, the classification is pretty much similar if you use biomarker data versus cognitive data. And you could say, well, then why not just go with cognitive data, because this is less invasive for the patients, is less expensive. Although asking clinical colleagues, they assured me that actually training nurses to take the clinical scores and the scales takes a lot of time and effort, so it is still expensive. But one could think, with technology like, for example, digital technology, patients can be at home, with some help from carers, could complete the cognitive tests on an iPad or any device. So the trick, though, is when we go on to the looking at the trajectories, actually the biomarker data helps more than the cognitive data. So although we can make the initial classification for a potential diagnosis with high accuracy, when we start looking at individual trajectories, the more biomarker data we have, the more precise these predictions become. So I think we don't yet have answers on what's the best data. And the interesting thing is, as you said, for example, UK Biobank has fantastic imaging data, but their cognitive profiling is not as deep as one would like. And other cohorts might not have state-of-the-art imaging data, but have a lot of cognitive profiling, a lot of clinical scales. So I guess we don't yet have the answer of what's, if you want, the absolute necessary and best assays to collect data from. And that's something we should be answering by putting our heads together in this space. But there is also another route that we can go down to, and I know a lot of people are working in this space, which is built up more a transfer learning type of algorithms that can bring a lot of data across different cohorts together, and harmonizing the data, starting kind of at the level of curating data, but also ensuring that the data can be harmonized through algorithms that allow us to actually learn biologically relevant, clinically relevant features, that random features that then reduce or increase the risk of non-generalizability across. So there are different approaches that we can go in terms of answering this question, but it is the key question. People ask us all the time within the NHS, well, so which data should we be collecting? Because when it will come to put this into a health check, people are not going to want to, be monitored the whole time by all sorts of devices, and they are not going to want to come back for many tests. And so practically, we need to answer this question soon.
And do you think that there is any opportunity kind of in the heterogeneity of data sources to answer as to which is the more the more efficient use of time or of imaging resources. Like, for example, ADNI is relatively heterogeneous, but it isn't entirely. So perhaps you could say, well, this protocol is actually acquired in half the time as that protocol, but we can get just as good a prediction out of it. Are those questions currently asked, or is this something that?
Yeah, I think they're really interesting questions, and it's a way that the different hackathons are now going towards answering. So it's quite, I'm sure everybody appreciates the challenge of comparing different models, but actually comparing this, if you want the same task on different data, could actually really help us answer these questions. Yeah. So do we need, the extensive imaging, or one type of imaging, could it be good enough? The other, if you want, kind of hope in a way, is that we could use a lot of the data currently in our hands through many research cohorts to build the models. But in the future, if these models are proven to be robust and generalizable, we don't need to be collecting the extensive data, biomarker data from all the patients. Maybe what we would need is the least kind of invasive structural scans. Maybe what we will see is that one functional, a 10-minute functional scan, like the resting state scan in biobank, together with a structural scan, could actually work perfect for new patients, as long as the models have been built as robust engines from all of the kind of large plethora of data that we can bring together. But first, we have to bring this data together, and we have to make these comparisons, as you're saying, to be able to answer. I might have it might have been a problem on my end. I do get a warning. I missed part of your answer in the end, but I think that was on my end. So please, if you do have a question on this or another aspect of the talk, please do put them in. Another question that I had was, you didn't, I don't think you touched upon the etiological interpretation side, but to what extent can we really learn new hypotheses as to how these neurodegenerative diseases actually work? Because clearly we don't entirely understand that yet. To what extent can we learn new hypotheses from this work? Yeah. This is also really interesting, and it's not just about prediction, it's about actually understanding the mechanisms of the disease. And what we see is in a way in two domains. One of the first things we learn is the interaction of different factors that can make a big difference. So, for example, there is work now showing that you can have a genetic high risk for dementia, but that can be mitigated by lifestyle factors. And that's only when we look at these large-scale data with machine learning algorithms that allow us to interrogate these very kind of complex and intricate relationships between the data that we start learning that how lifestyle factors interact actually with biology, with genetics, with pathology. And that's something really, really important. It has practical application, but it's also really important in terms of understanding the causes of disease and the etiology. So that this is one space. Another space where we have a lot to learn is the different subtypes of dementia. And this, again, has great clinical relevance in terms of patient pathways and in terms of drug discovery, but it's also really important for understanding the mechanism. So we see from recent machine learning approaches that new subtypes emerge when the algorithms interrogate, again, these interactive factors. We start seeing clusters of patients that don't fit with the clinical diagnosis they were given. And so who are they? What are their symptoms and what is their pathology? And we start building kind of new models of disease pathology. So I think it's really a fascinating area because it requires people coming together at the interface of clinical practice and knowledge, neuroscience, obviously, machine learning space. So there's really a lot to learn about disease in this space and understanding the mechanisms. And of course, only then we will be able to have the breakthroughs in the more kind of translational domain that are very much needed for treating the disease.
Thank you very much. Massively fascinating topic. Thank you very much for an excellent talk. That brings us to the end of the session. I'd like to hand back to Carola for some closing remarks. Thank you.
Thank you so much, Marius. Thank you very much. I'll also invite James and John to join me for the closing remarks if they want to. I really had 2 very, very good days. It was super inspiring, very interesting talks. Thank you so much to all the speakers for contributing. Thank you to the audience for being so engaged and for attending, for participating, for asking questions. And also thanks a lot to the Newton Gateway and to Josh for really perfect organization as always. Thank you so much for helping us with that. I think what we have really beautifully seen is this interplay of mathematics, statistics, machine learning, that we've really seen that at work in the presentations that have happened. And we've also seen the really huge potential that these type of approaches have to really make a difference and to support clinical practice. So there is a lot that we can do already. There is a lot more that we are hoping to be able to do in the future. And this is also part of what our hub's mission is. And so there is much more to come. So please stay tuned. There are more events coming up of the CMIH. And we are very happy to discuss more, to engage, to collaborate. Thank you very much. I'll hand over to James and John now.
Yeah, just to echo those comments, it's been a fantastic meeting. Please do contact us if you have ideas for collaboration, particularly clinicians in the audience, biomedical researchers. We're always keen to hear from new areas of medicine where ML is underrepresented. And yeah, thank you so much for your time, and thanks once again to the great speakers and organizers of the event. Look forward to hearing from you guys in the future.
Yeah, I don't think I can add more other than to say it's been an excellent couple of days. I've learned so much, and I'd just like to thank everybody again for such a splendid organisation and the talks, and everyone for participating so well.
Thank you, everyone.